So now we're going to focus on genetics and inheritance. Inheritance is all about passing on our genes from generation to generation, passing on certain characteristics, certain traits from generation to generation. And the study of the genes and how they are inherited is what we call genetics. So, what is a gene? A gene is the biological, or you can say the functional unit of heredity. Like I said before, it determines the traits of an individual. It basically is a sequence of DNA that encodes the information to make proteins. So remember that all proteins within the body are actually made from a DNA sequence. A DNA sequence contained the instructions or had the blueprint, right, of making that protein. So, what is a chromosome then? A chromosome consists of a long strand of DNA that contains multiple genes. So the chromosome is a tightly coiled form, a tightly coiled structure of the DNA. It's usually visible during cellular division. So what are homologous chromosomes? So first, let me say this. For us humans, we have 46 chromosomes. Now, we received each set, we basically inherited 23, a set of 23 chromosomes from our dad and a set of 23 chromosomes from our mom to give us the 46. Are you following? Now, these sets of chromosomes are actually called homologous chromosomes. Homologous chromosomes. So homologous chromosomes carry the same, basically, they carry the same gene. Let's put it that way. They carry the same gene. Although they carry the same gene, they may carry different versions of the same gene. What am I trying to say? For example, the gene for eye color, I inherited the gene for eye color from my mom and my dad. But I am carrying two versions of the genes. So imagine if your dad had blue eye color, but your mom has green eye color. So those are two versions of the gene for eye color. Are you following? And you inherit both. You do. But the one that is expressed, the one that you show physically, will depend on the nature of the inheritance pattern, which I will talk about very soon. All right, so just remember, homologous chromosomes are chromosomes that carry the same genes at the same location to determine the same traits, such as eye color, but they contain different alleles. So an allele is basically a different version of a gene. Okay, so several terms that you should know: alleles, different versions of a gene. Okay, we already covered what a gene is. We already talked about homologous chromosomes and what a chromosome is. Now, let's go back to the homologous chromosomes for a second. Remember what I said, that we had 46 chromosomes, right? Basically coming in 23 homologous pairs. Now, 22 out of the 23 pairs, the 22 pairs of chromosomes are actually called autosomes. Autosomes are known as autosomes. The autosomes have nothing to do with your sex. Has nothing to do with you being a male or a female. They're found in all of your body cells. Okay? But the 23rd pair of your 23 pair of chromosomes, that 23rd pair is what we call the sex chromosomes. They usually determine your sex. So if you're female, your 23rd pair will be X and X. If you're male, your 23rd pair will be X and Y chromosomes. Okay, so remember that we have 23 homologous pairs. The first 22 pairs are called the autosomes, and the last pair are the sex chromosomes that determines our, sex, if we're male or female. Again, alleles are basically different versions of a specific gene. As a result, we can designate alleles as dominant or recessive. The dominant alleles are going to mask the effects of other alleles. They will suppress the effect of other alleles. The dominant alleles, they will be expressed physically in the individual. Okay? Recessive alleles, they're the ones that are suppressed. They're the ones that are basically masked by other alleles. They're usually not expressed at all times, but there is a rule. We do have a condition that will enable a recessive allele to be expressed physically in an individual, and this is a rule. Here we go. For a recessive allele to be expressed physically in an individual, you need 2 copies of that recessive allele. So what does that mean? It means you inherited the recessive alleles from both your mom and your dad for you to show it physically. Are you following? Now, what about the dominant allele? You just need 1 copy to express the dominant allele physically. Okay? Now, the dominant allele is usually represented by an uppercase letter, any uppercase letter, and the recessive allele is represented by a lowercase letter. As a result of this, we can come up with different combinations of a genotype. So a genotype is the individual's genetic makeup. It basically refers to your combination of alleles inherited. So for example, if you inherited 2 dominant alleles from your parents, you will be a homozygous dominant individual for that particular gene. If you inherited a dominant allele and a recessive allele from your parents, you are a heterozygous individual for a particular gene. Now, if you inherited 2 copies of the recessive alleles from mom and dad, then you are a homozygous recessive individual for that particular gene. Okay, so the genotype is your genetic makeup. It's the combination of your alleles that you inherited from your parents. Now, what is a phenotype? The phenotype is the outward appearance. It's that physical expression of the genotype. Okay, so the phenotype is dependent on the genotype. All right. So this means a homozygous dominant individual, if their genotype is homozygous dominant, externally or, physically, they will express the dominant trait. Same thing with a heterozygous individual. They will express physically the dominant trait. The reason is because you only need 1 copy of the dominant allele for you to show it physically. Okay? However, the homozygous recessive genotype will produce the recessive trait physically in the individual. And we have several Punnett square examples to use as we go along today. So now we're going to cover different types of inheritance patterns that are associated with the autosomal chromosomes. So remember that these are associated with the autosomal chromosomes. And then we have another group of inheritance patterns that are associated with the sex chromosomes. So we have the strict dominant and recessive inheritance patterns where certain traits, are found or associated with the dominant allele, while other traits are associated with the recessive allele. We also have the codominance inheritance pattern. And this is where two alleles are codominant to each other, meaning they do not suppress each other. So one is not dominant over the other. Rather, both are expressed physically in the individual. Perfect example of this is the ABO blood group, and we will cover this in depth when we do examples of Punnett square questions. And then we also have the sex chromosomes. Now, inheritance patterns associated with the sex chromosomes, we call this sex-linked inheritance, sex-linked inheritance. So these traits or, disorders or characteristics are associated with either the X chromosome or the Y chromosomes. And examples of these are color blindness, the red-green color blindness, hemophilia, and the Duchenne's muscular dystrophy. So how can we determine the probability of an offspring expressing a particular, genotype or phenotype? How can we determine the possibility of an offspring inheriting a genetic condition? What can we use? What do geneticists use in determining these probabilities? Well, we can use a Punnett square. So a Punnett square is very helpful in determining the different inheritance of dominant and recessive traits, and also traits that are associated with the sex chromosomes. So we will begin with this first Punnett square problem. So here we have long hair is dominant to short hair. So long hair is a dominant trait. So we know that. So we can represent the long hair with an uppercase letter. Any letter will do. And we can represent the short hair with the lowercase version of that letter. So what are we doing here? It is asking for the probability that the offspring will have long hair. What is the probability the offspring will have long hair if we have a mating between a heterozygous female and a homozygous recessive male? So I would recommend that you first choose a letter and then use that letter in its uppercase form to represent the dominant trait and its lowercase form to represent the recessive trait. And then determine the genotype of the female and the genotype of the male. So the answer is 50%, but how was that derived? So here it is. So first, like I said, determine your letter. You can choose any letter that you would like. Okay. And then once you've chosen that, the uppercase letter will be for the long hair, which is the dominant trait. And the lowercase will represent the short hair. And then go ahead and write down the genotype of the woman and the genotype of the man. And it is possible to do that quickly because the female is heterozygous. Heterozygous means different, so it means we're going to have a dominant and a recessive allele combined. Now, homozygous recessive means we have two recessive alleles, and that's what we see here. And then you go ahead and draw the Punnett square. 4 boxes, so you can put the man at the top, the genotype of the man at the top, and the genotype of the woman on the side. Doesn't matter. You can switch it. That's okay. And then you go ahead and cross it. So, of course, we have the uppercase, the dominant allele going with the recessive allele. Same thing here. And here we have two recessive alleles and two recessive alleles. So you complete the Punnett square and then interpret the Punnett square. So looking at this, in the first box, we have the dominant allele being expressed. Notice there is one copy of the dominant allele, so it will mask the presence of the recessive allele. So this individual here, this will be long hair. They'll have long hair. How about this one? Same thing here. It's going to be long hair here. Okay, so long hair and long hair. Now, for these two, we have two copies of the recessive allele. Remember, if we have two copies of the recessive allele, then the recessive allele will be expressed. That trait will be expressed. So here we're going to have short hair and short hair. So of course, the chances of the offspring having long hair, it will be 50%. Okay? Here is a second problem. You can do the same thing with the second problem. I'll let you work through that. The answer key is embedded within the presentation. Please work through it. Now, in this problem, it tells you that neurofibromatosis is a genetic disorder, is an autosomal dominant disorder. Autosomal dominant disorder. So first, it's telling me that this condition is associated with the dominant allele. It's a dominant trait. So this has nothing to do with the sex chromosomes. This is the autosomal chromosomes, but this is a dominant trait. So an individual that has the dominant allele will most likely have this condition. So that's my interpretation from just reading this first sentence right there. All right, now let's take a look at the genotypes of the man and the woman. So the genotype of the man is, he is heterozygous. And the genotype of the woman is, she is homozygous recessive. So you can choose any letter. So I would say the man is heterozygous. So we can use the letter B, and I would represent the man like that. So this is the male. Now the female, she's homozygous recessive for the disorder, so I would give her 2 recessive alleles. So this is the first thing you should do. Define the genotypes, okay? Define the genotypes. When you're reading the information on the question stem, define the genotypes for the individuals before you set up your Punnett square. Okay, now the question is asking for what is the probability that their offspring will be, will have neurofibromatosis. So what is that probability? Well, draw out the Punnett square, go through it, and come up with your answers, okay? So I hope your answer was D. It is 50%. And here we have the explanations for you, all right? Here is another one. So in this case here, we have albinism. And here it's telling me that it is an autosomal recessive disorder. So it is recessive. So it is attached to the recessive allele. So if I inherit 2 copies of the recessive allele for this condition, I will most likely have this condition. Remember, it is the rule. For the recessive allele to be expressed externally, you need 2 copies of it. You should have inherited 2 copies. For the dominant allele to be expressed physically, you need just 1 copy. Okay, so now again, go through, read the question stem, determine the genotype for the man and the genotype of the woman. So the man's genotype is, he's heterozygous. So go ahead and write that down. Choose any letter that you would like to use and write down the genotype for this heterozygous male. And then write down the genotype for this homozygous recessive female. Okay? Do the Punnett square and determine what percent of their offspring will be albino. Right? So I hope you came up with D. It's going to be 50%. Now you can walk through the answer key here. I hope this makes sense to you. If it doesn't, definitely see me during our class sessions to help you go through this questions, okay? Now, the second inheritance pattern that we're going to talk about is codominance. Codominance. And the perfect example of this one is the ABO blood group. So our blood types are really determined by 3 different alleles in the population. We have the A allele, the B allele, and the I allele. The A allele usually codes for the A antigen that sits on the membrane, the surface of the red blood cells. The B allele will code for the B antigen. And the I allele, it codes for no antigens on the surface of the red blood cells. However, the I allele is recessive to both the A and the B allele. But the A and B allele are dominant over the I allele. Okay, and I will explain why in a second. So if you inherit, let me say it this way, if you inherit the I allele and the A allele from your parents, your blood type will definitely be type A. If you inherit the I allele and the B allele from your parents, your blood type will be type B. Remember, the A and B allele are dominant over the I allele. Now, what if you inherit the A allele and the B allele? What will be your blood type? Your blood type will be AB. The reason is because both of these alleles are co-dominant. They are both expressed. One is not dominant over the other. They're both expressed physically on the red blood cell surface. Okay? So now how do we represent this information genotypically? Let's take a look at this chart. So if your blood type is A, if you're type A, that's your phenotype. So your blood types are the phenotypes. If you're blood type A, that's your phenotype. Your genotype can be the following: homozygous for blood type A, or heterozygous for blood type A. So the heterozygous form is basically the type A plus the O. So that's the heterozygous form. So keep that in mind. If you're blood type B, your possible genotypes are homozygous for blood type B or heterozygous for blood type B. All right? Now, if you are type AB phenotypically, the only possible genotype for you is AB. Same thing with the phenotype O. The only possible genotype for you will be OO. So using this information, we can use a Punnett square to predict the chances that an offspring will inherit a specific blood type. So here's a Punnett square question, and we can go through it together. So here, a man who is heterozygous for the blood type A. So I'm expecting his genotype to be AO. He's heterozygous, different. So AO. While his wife is homozygous for the blood type B. So his wife, her genotype is BB. Okay, so what is the probability their offspring will have blood type B? So you go ahead and take a second and work through it. So the answer is 50%. And And this is how it was generated. So using the Punnett square, here we are. So here are the possibilities. So I can see that there's a 50% chance, there's a 50% chance right here that the offspring will have the blood type AB. Notice that. See that right there? And there is also a 50% chance that the offspring will have blood type B. So don't forget, these are the genotypes, but when you go to write the phenotype, which is the blood type, then you ignore the O. Okay? So it's a 50% chance. So here's another one right here. So here the blood type, the man is homozygous for the blood type A. The wife is also homozygous for the blood type B. So again, write down the genotype. So for here, it's going to be A, and here it's going to be B, B. Okay, so let's go back to that. So what is the probability that their offspring will have blood type AB? Well, you draw the Punnett square. So when you draw the Punnett square, I like to put the man here. So A, and the woman will be B, and B here. Okay, so it will be AB and AB. So if you notice, it's going to be a 100% chance that their offspring will inherit or have the blood type AB. Okay. Now we're going to focus on sex-linked inheritance. So these are inheritance patterns that are associated with the sex chromosome. So the traits are usually found on either the X chromosome or the Y chromosomes. Now, X-linked traits are very common, or X-linked disorders are very, very common. So we're going to do some examples of some Punnett square questions related to sex-linked inheritance. So to determine the Punnett square, or to draw the Punnett square, you first need to determine certain terms. So if you're saying a condition is X-linked recessive, what you're saying is that the recessive trait is linked or associated, with the X chromosome. So for an individual that is hemophiliac, for example, the female, remember the female has two X chromosomes and the male has one X chromosome and a Y chromosome. So how would you represent it if you're dealing with a hemophiliac female? You would represent it this way, and they're telling you that the disorder is recessive, X-linked recessive. Remember, for you to have it, for you to show the recessive trait, you must have two copies of that recessive allele. So sure enough, it is right here. I can see the two copies of the recessive alleles attached to the X chromosomes. Now, what if it's a hemophiliac male? Well, they only have one X chromosome, so that X chromosome will carry the recessive allele. So this is how you would represent it when you're trying to define the genotypes for the female and the male. So here's a Punnett square question that we can do. So color blindness is X-linked recessive. Okay, so for someone to be colorblind, if it's a female, they need two copies of the recessive allele. If it's a male, they just need one copy because they only have one X chromosome. They need one copy of the recessive allele. So the question stem says, if a colorblind woman and a normal vision man have a son, what is the percent chance that he will be colorblind? So the female, she is colorblind, so she is carrying two recessive allele. So you would put, to set up the Punnett square, you have to use X and X for the female and X and Y for the male. Okay? Now for the man, he has normal vision, so we will give him the dominant allele right there for normal vision. Okay? And then you go ahead and cross the genotypes, and here are the results. So we can see over here, we have a normal vision female. And here we also have a normal vision female. But over here, I see that we have a colorblind male and a colorblind male. So what is the percent chance that their son, please pay attention to this, their son will have color blindness? So if you're focusing on the sun, you just have to focus on this box here, this region, is this part. Okay, and notice it's a 100% chance that their son will have color blindness. Right? Here's another one. Let's try this one. So here again, we're dealing with color blindness, X-linked recessive, but this time the woman is heterozygous for the trait. So she has a dominant and a recessive allele, and we can see that here. Okay. Now she's married to a colorblind man. So he has the recessive allele. He only needs one because he has just one X chromosome. What is a percent chance that their female offspring will be colorblind? Well, here it is. Notice once you're done crossing, let's take a look at this female here. This female here will have normal vision, but this one will be colorblind. Why? They are carrying, this one is carrying 2 of the recessive allele. So this female will be colorblind. How about on this side? This male will have normal vision, but this male will be colorblind. But the question is asking for what? What is the percent chance their female offspring will be colorblind? Well, we will focus on this area right here where we have the XX chromosomes. So just this part. And if you notice, it's only a 50% chance that the female, so we're just focusing on this area, and if you notice, half will have normal vision. The other half will be colorblind. So it's a 50% chance that their female offspring will be colorblind. So I encourage you to go through the Punnett square questions. Be prepared to do multiple questions, such as the ones that I have gone over with you on the exam. So now we will focus on genetic screening. So how do geneticists, assess the likelihood that you will inherit a specific genetic disorder or a specific trait in the family? Well, there are different ways that we can do it. We have pedigree analysis. We can use karyotyping. Now, if we're talking about the fetus, we can use immunosynthesis, chorionic villi sampling. So we're going to go through all these. So the first one will be pedigree analysis. Now, a pedigree can be constructed from your family history, and using that, it can determine your likelihood of inheriting a genetic disorder or a specific trait. The other one is karyotyping. Now, karyotyping, first, what is a karyotype? Let's define that. A karyotype is actually a visual representation, and you can see it right here. It's basically a picture that shows the number of chromosomes within an individual. It shows you the banding patterns, the length of the chromosomes and their positions, and it shows you if a chromosome is missing or if all is well and if all the numbers of chromosomes are there. So this is why karyotyping is very important. So if we have abnormalities such as, non-disjunction, where we're missing one chromosome, we can see this, using the karyotype. We can see that. So if an X chromosome is missing, we can see that. If it's a female, we can see it on a karyotype. So karyotypes are very important. But before we can do the karyotype, we usually take the fetal cells, if we're dealing with a fetus and we're trying to figure out if this fetus will inherit a specific condition, for example, Down syndrome. So if a mother wants to check and see if her, her child, her fetus will have Down syndrome, they will actually take samples of the fetal cells and use it for karyotyping to determine if there are missing, extra, or mutated chromosomes. And one of the process of getting these fetal cells during pregnancy is what we call amniocentesis. So this is when a small amount of the amniotic fluid is taken, during the 14th through 16th week of pregnancy, and that little amount of amniotic fluid is then tested. And once it is tested, they can run it, through multiple lab processes, and of course karyotype it to show us the chromosomes and check for any abnormalities. The other process is the chorionic villi sampling. So they take a sample of the chorionic villus tissue during pregnancy, and then of course they'll process that in the lab, and then they're going to karyotype those cells, and it will reveal if we have any abnormalities with the chromosomes of the fetal cells. So this is how they get the fetal samples, and then of course do the karyotyping to determine the likelihood that an individual will inherit a genetic disorder or a specific trait. Finally, we have the genetic disorders, and for this segment of your lecture notes, I want you to take the time to study this. Okay, I'm not going to lecture through this. I want you to read it. It's in depth. You don't have to know all the specifics. I basically just want you to know an overview of these genetic disorders. Now we have non-disjunction, and non-disjunction disorders are usually due to the failure of the homologous chromosomes or the sister chromatids, their failure to separate during meiosis. Okay, so that is how non-disjunction disorders come about. And then, of course, we have the autosomal dominant disorders, the autosomal recessive disorders, and then the sex-linked disorders. We've mentioned a couple of them. So go through them, be familiar with them, and be prepared for questions on the exam.